Making proteins. In this lesson, we'll look at the science and engineering practices of developing and using models, constructing explanations and designing solutions, as well as obtaining, evaluating, and communicating information. Our cross-cutting concepts include cause and effect, scale, proportion, and quantity, systems and system models, as well as structure and function. Introducing proteins. Proteins are a very diverse group of biological molecules made up of long chains of amino acids. They have a wide range of biological roles. Those roles include structural roles. Proteins are the main component of most of our body tissues. They have catalytic roles. All enzymes are proteins, and these catalyze many biological reactions that occur in our bodies. They speed them up or they slow them down. They are used for signaling. Many of our hormones and cell receptors are proteins. And finally, immunological. All of our antibodies are proteins. The role of DNA. As we start talking about protein production, we can't forget about DNA. DNA is a long molecule found in the nucleus of cells. It determines the function of cells by controlling the production of proteins. Short sections along the DNA molecule contain the information required to make proteins. These sections are called genes. All of your body cells contain the same genes. Active and inactive genes. Many of the genes inside any particular cell are inactive or switched off. Inactive genes do not produce any protein. The active genes control the activity of the cell by making sure it produces the specific proteins it needs to function properly. Whether a gene is active or inactive depends on the type of cell it is in. This means that different types of cells produce different proteins. For example, red blood cells produce the protein hemoglobin. White blood cells produce antibodies. The triplet code. DNA is a polymer made of a chain of nucleotides. Each nucleotide contains a base. There are 4 different types of bases represented using the letters A, T, G, and C. A stands for adenine, T stands for thymine, G stands for guanine, and C stands for cytosine. In order of bases in a gene determines the order of amino acids in a protein. A set of 3 DNA bases corresponds to a single amino acid. This is called the triplet code. So we've got our sequence of 3 base pairs. This is our triplet code of 3 bases. It is important that the correct order of amino acids is achieved in order for each protein to function properly. So what is the link between genes and proteins? The genetic code controls cell activity by coding for the production of specific proteins. DNA and the bases. DNA contains a long sequence of chemicals called bases. The sequence of bases provides the genetic code for the production of proteins. Pairs and triplets. Bases on opposite strands of the DNA molecule link to form base pairs. 3 base pairs make up a triplet. Triplets and amino acids. Different triplets code for different amino acids. The order of triplets determines the order of amino acids. Each protein is made of a different sequence of amino acids. So here we have the triplet TTT. It codes for the amino acid number 1. The next triplet, CTG, codes for a completely different amino acid. The third triplet, GTG, codes for yet a different amino acid. And CCT codes for another amino acid. So how are proteins synthesized from DNA? This process is called protein synthesis. It involves the production of a chain of amino acids called a polypeptide chain. The stages of protein synthesis include: number 1, transcription of the gene in the nucleus to make a strand of mRNA. And number 2, translation of the mRNA on a ribosome to form a polypeptide chain. Finally, 3, modification of the polypeptide chain to make the final protein. So let's delve into this a little deeper. Transcription in the nucleus. Protein synthesis begins in the nucleus. The DNA is too large to move into the cytoplasm, and so a copy of the gene must be made. This is called transcription, and it involves a number of steps. The 2 strands of DNA must be unzipped or separated to allow access to the sequence of bases in the gene. There are specific enzymes that allow for the unzipping of a strand of DNA. A large protein called RNA polymerase binds to a section of the DNA in front of the gene. RNA polymerase runs along the DNA, producing a copy of the gene. This copy is called mRNA, or messenger RNA. It is sending the message of DNA and allowing the ribosome to read that message and eventually make proteins. Stage 2, translation in the cytoplasm. Following transcription, the mRNA strand moves out of the nucleus and into the cytoplasm, where it attaches to a ribosome. The mRNA acts as a template for translation. A carrier molecule called transfer RNA, or tRNA, brings amino acids to the ribosome. The ribosome reads the mRNA and joins the correct amino acids together according to the triplet code. When the final amino acid is added, the complete polypeptide chain is released into the cytoplasm. So continuing to look at how proteins are made. Each gene in the DNA molecule codes for a different protein. Genetic material exists in the nucleus, but protein synthesis occurs in the cytoplasm, where it is carried out by ribosomes. Therefore, the gene must be copied. Genes are in the nucleus, but protein synthesis occurs here at the ribosome. These genes and the DNA that exists here cannot leave the nucleus, and so an mRNA strand is made that is small enough to leave the nucleus and be taken up by the ribosome. Stage 1. The section of DNA containing the gene unwinds and the 2 strands separate. This exposes the bases on each strand. Stage 2. a molecule of RNA is made using the exposed bases as a template. This is called transcription. Stage 3, the new RNA molecule is called messenger RNA, or mRNA. When it is complete, the mRNA moves out of the nucleus and into the cytoplasm. Stage 4, the mRNA attaches to the ribosome in the cytoplasm. The ribosome starts to read the sequence of base triplets in the mRNA. As each triplet is read, the correct amino acid is added to the growing chain by tRNA, or transfer RNA. The amino acids link up to form a polypeptide chain. Here's our polypeptide chain. One amino acid is assembled, another one is assembled after, and then another is assembled after. This is called translation. We are translating the information in the mRNA, and it is being turned into a polypeptide chain of a number of different types of amino acids. Remember, your proteins are made up of amino acids. Stage 6, when the last triplet of the mRNA is read, the complete polypeptide is released into the cytoplasm. The final protein may contain several polypeptide chains. Stage 3, formation of the final protein. The final polypeptide chain produced during translation must fold into a unique three-dimensional shape. This allows it to carry out its specific function. So here's our polypeptide chain, and as it is folded, it becomes the shape that the protein needs. These shapes allow for reactions to take place. For example, an enzyme must fold correctly to form its active site. This enables it to bind to substrates. A protein can be made of more than one polypeptide chain folding together. For example, the protein collagen is made up of 3 polypeptide chains. So let's review the sequence of events in protein synthesis. Number 1, a section of DNA unwinds and the 2 strands separate. Number 2, the gene is copied into a strand of mRNA, and this is transcription. Number 3, the mRNA leaves the nucleus and enters the cytoplasm. Number 4, the mRNA attaches to a ribosome. Number 5, the ribosome reads the sequence of bases in the mRNA. Number 6, transfer RNA, or tRNA, carrying amino acids, builds a polypeptide chain. This is the process of translation. Number 7, a completed polypeptide chain is released from the ribosome. And number 8, a protein is formed from several polypeptide chains. Changing the protein sequence. Each protein has a specific number and sequence of amino acids that determines its shape and function. If just one amino acid is incorrect, the protein might not function correctly. Changes to the order of bases in DNA can alter the order of amino acids in a protein. These changes in DNA are called mutations, and they can be due to the deletion of a base, the addition or insertion of an extra base, or the replacement of one base for another. Mutations are continuously occurring in your body. However, most do not affect protein function. This is because many mutations do not alter the protein, or do not alter it significantly enough to affect its function. Some mutations can be beneficial and provide the genetic variation needed for evolution. For example, a mutation may change the phenotype of an individual. Recall that the phenotype is the physical characteristic that is expressed. This might provide them with a greater ability to survive. Those organisms that survive are more likely to reproduce, and so those genes are more likely to be passed on to future generations. Mutations in coding DNA. Coding DNA is part of a gene which determines the order of amino acids in a protein. When a mutation occurs in coding DNA, it can alter the phenotype of an individual by changing the activity of a protein. A change in the base sequence of a gene may lead to the production of a different protein. A change in the protein structure, or no protein being made at all. So here we see an altered protein activity. This protein initially was here, and now it becomes this. This would cause this entire polypeptide chain to fold differently into a protein that is not like the previous protein. Mutations and enzymes. So what could happen if a mutation occurs in a gene coding for an enzyme? Remember, enzymes speed up chemical reactions. If a mutation alters the structure of an enzyme's active site, the substrate, the material that binds to it, may no longer be able to fit in the enzyme's active site. This will prevent it from catalyzing important cellular reactions that enable the body to function. Mutations that affect other parts of the enzyme may alter the location of the enzyme in the cell, which could affect its activity. Mutations do also occur in non-coding DNA. Some sections of DNA do not code for proteins. This is called a non-coding DNA. Some non-coding DNA can control whether a specific gene is transcribed by switching it on or off. Mutations in non-coding DNA can affect the phenotype of an individual by changing gene transcription. This is because RNA polymerase binds to non-coding DNA at the start of transcription. So what will happen if a mutation in a non-coding DNA prevents RNA polymerase from binding? Potentially the entire gene where the binding site is taking place may not be coded for. So an entire protein might not be developed. Continuing with mutations in non-coding DNA. Mutations in non-coding DNA at the start of a gene can prevent RNA polymerase from binding to DNA. If RNA polymerase is unable to bind to the DNA, then transcription cannot take place. This results in a decrease in mRNA production, which means less protein is made during translation. Mutations in non-coding DNA can therefore affect the amount of protein produced in a cell. Mutagenic agents. Most mutations occur when DNA is replicated in cell division. During cell division, a new cell containing an identical copy of DNA to the parent cell is produced. Sometimes mistakes are made when the DNA is copied, leading to mutation. The occurrence of mutations can be increased by mutagenic agents, such as UV radiation and harmful chemicals. Mutagenic agents disrupt the structure of DNA or interfere with protein synthesis.